Attendees are in listen-only mode.
Hello, and welcome to Transposagen's webinar on footprint-free cell line engineering. I'm Jack Crawford. I'm the Director of Sales and Marketing at Transposagen, and we appreciate your interest in our technologies and services, and thank you for joining us today. A few points before we begin. This webinar is being recorded, and we will send all registrants a link to the recording in case you couldn't make it, as well as a PDF version of the slide deck. And feel free to send these to any colleagues or people of interest. The webinar system allows you to type in questions on the panel on the right towards the bottom. We encourage questions throughout the webinar, and we will answer a couple questions at the end of the webinar, as well as address all questions directly with you with our scientific staff. With that, I'd like to introduce our presenters today. Our main presenter is Ken Miller. He's a senior scientist at Transposagen. Ken got his master's degree from the University of Illinois. Previous to Transposagen, he worked at Schering-Plough and Pfizer. Ken has 20 years of experience in cell line engineering, design, and production. Ken is also responsible for hundreds of successful gene editing projects at Transposagen, both internally for our projects and with our clients. Also with us today is Valeria Steffi, and she is our cell culture expert as well as transfection specialist. Valeria has 15 years as a practicing medical doctor, 9 years in neuroscience research, and 3 years experience with stem cell culture and manipulation. She recently completed the human IPS training course at the NIH as well. A little bit about Transposagen. Transposagen is a gene editing company that was founded about 10 years ago with the original goal of being the first and the best at creating genetically engineered rats. And throughout obtaining that goal, Transposagen in-licensed and developed the expertise for a number of different gene editing technologies, including the XTN TALENs site-specific nucleases, CRISPR-Cas site-specific nuclease and nickase systems, the piggyBac DNA modification system and transposon, as well as stem cell modification systems. And since obtaining that goal of being the first and the best at genetic modification in rats, we've expanded our services and our products to create custom gene editing tools or reagents and deliver those directly to the client or to the researcher to use for their specific application in their lab or within their company, as well as we've created services for custom cell line engineering and custom stem cell engineering. Today we'll be focusing on footprint-free gene editing in cells, and Transposagen has an exclusive license to patents and have developed our own patent portfolio for the piggyBac transposon system, both for hyperactive transposases as well as excision-only transposases. So we are truly the only commercial source for footprint-free gene editing and clean genome engineering. And with that, I'm going to turn this over to Ken Miller, who will get into the technical portion of this webinar.
Okay. Thank you, Jack. Hello, everyone. I want to thank you for logging on and joining us today. We're excited today to tell you about the cell line engineering service we now offer at Transposagen. And as Jack mentioned, in reality, we've been at this for quite a while with our in-house projects, and I realize that we've assembled the tools and capabilities and expertise in-house to offer this service to the research community. And so I'm going to go over some slides that outline these capabilities, including some examples from our in-house research. First slide I've got up here is just, again, an overview of what the types of cell projects that we can offer, along with some of the technologies that are required to perform those projects, and You can see in the middle, a lot of these projects involve either TALENs, which we call our TALENs XTNs, but I'll refer to them as TALENs throughout the talk, or CRISPRs or nickases and so forth, as well as the piggyBac transposase that Jack mentioned. So before we get into too much, let me back up just a minute and just quickly review what happens when a cell gets a double-strand break. As many of you probably know, one outcome is what's known as non-homologous end joining. So if there's a double-stranded break in the DNA, often the repair is error-prone, and you can introduce small deletions or insertions, referred to as indels. This is a common outcome, and this is something we can use to our advantage if we want to knock out a gene. But if you give the cell a repair vector with homologous sequences, then it can use that sequence to repair the double-stranded break in an error-free manner. So just to again see the various outcomes that can be achieved with this double-stranded break tools. Again, on the left is the non-homologous end joining, and you can see that in that case you might knock out a gene by throwing it out of frame if the repair is not an in-frame repair. And so you can target an exon close to the front or in an area that you know to be important in the protein and knock out a gene that way. But again, in the middle and right frame, if you give it that repair molecule, you can do a couple of things. You can either knock in a larger piece of DNA, as is shown in the middle, where you might knock in a gene or a cDNA or a portion of a cDNA or an exon. And then on the right, you might also just give it enough sequence to do a very small but precise editing of that sequence, maybe a couple of base pairs, one base pair, and repair the sequence in that way. So with this in mind, with this little bit of background, one of the types of projects that we offer and have done already for a customer is just the knockout of a gene in a transformed cell line. So this is an example of one that we did in CHO cells, which, as you know, is a very important commercial cell line. And these type of cells are amenable to multiple rounds of transfection and cutting, since they're so robust and are immortal, of course. And so what kind of a workflow might look something like the inset box there, where you would design a couple of talons, and we have the capability to make them here, so we can make as many as we need. But you pick a exon again that you think is important or know is important from the literature, transfect your talons in there, screen for activity or for cutting by some method, either maybe a qPCR probe or whatever your method of choice. And then if you see that level higher than you would like, you hit it again, hit the cells again. As everybody knows, talons are very precise in their cutting, and so you can do this 2 or 3 times, screen for activity, and then once you feel like you've got the activity or the activity of your probe low enough, you then go clonal and do the follow-up sequence analysis like is shown here. And this is just to kind of, if you've never seen an indel that you might get from a TALEN or a CRISPR cut, you just get various sizes of, usually deletions, sometimes insertions of various sizes. And this is an example of something we got here. Had a lot of success, and were able to deliver this product to the customer. They were very happy with it, and they're using it as we speak, as far as we know. So other than those kinds of projects, very often if you want to knock in a piece of DNA or a gene edit or something, very often you want to use selection. And so, as many of you know, if you've worked with cells at all, finding the cells that have the alteration that you desire is the tricky part. And so everybody always very often uses antibiotic selections, and we'll talk about some details there. But obviously there's, another alternative is to try to go with maybe a single-strand oligo method. So you would cut with a CRISPR or a TALEN and put in just an oligo. And there's literature on that approach. It's changing every day, of course, but one of the issues with that is finding a relatively rare gene edit that you're looking for. Since there's no selection, you have to kind of sort through a lot of clones, and B, it can most of the changes are monoallelic when you do find them, and if you're looking for a biallelic gene edit, this is even maybe a harder method to go with. And so our solution is to use footprint-free gene editing using the piggyBac transposase, which, as Jack mentioned, is something we exclusively can offer. And I'm going to go through some of the details of that coming up. So I thought we would just do a quick step through of what is involved in a typical footprint-free engineering project. And so obviously you got to design the vector, and I'll go through some nitty-gritty details of that in a couple of slides. Then you got to do the targeting, and select it using that antibiotic resistance that I mentioned, screen for correctly targeted clones. And then with our system, the next step is to remove that cassette back out of there, because you don't want to leave that in the genome. We use piggyBac, of course, as we've mentioned, and then select and screen for excised clones, and then follow up with clonal isolation and any further molecular analysis or sequencing you need to do to make sure you've got exactly what you want. So we use, obviously, various selections are available to us. We actually sell a vector called we call a multi-vector, and it's just various vectors that have different antibiotic resistances available, and or maybe a fluorescent marker in them as well. And you can see them listed there. A lot of people like to use Puros, a popular antibiotic, quick killer. But you may have an antibiotic already in your cells for whatever reason, and so you might need to choose something different. You'll also note that many of these vectors contain the TK gene, the thymidine kinase gene. Any of you from the mouse targeting world will recognize this as a tool that's used in a positive-negative selection scheme when you're targeting ES cells. And so in this case, it's used a little differently. We're going to use this to select for excision of our cassette. So we're not using it to select for proper integration. We use it later, and I'll outline how that happens in a couple of slides. And so you can see some of the features down below if you want to peruse those. But in general, it's a vector that's made for making homologous recombination vectors. Okay, so this slide, I'm going to go through. This is the nitty-gritty details slide that I mentioned before. But in general, often a customer will come to us and say, I'd like to make a single base change in particular exon of this particular gene, and I've kind of illustrated this as you see here with this. Hopefully you can see my pointer here. This pink G here, this is the G that they might want to change. And so what we do is look for the closest TTAA site. We haven't mentioned yet, but piggyBac transposase recognizes TTAA sequences in the genome and can insert transgenes into TTAA sites. And so, but we're going to use that the endogenous TTAA sequence to our advantage in this case, as you'll see in a minute, to place our selection cassette and our gene edit at that TTAA site, so that when we remove the cassette later, it'll be a footprint-free edit, and I'll show you how that works here in a minute. And so, importantly, if a TTAA site, so in this case, I've shown that we're going to put the selection cassette in an intrine, close to where we want our gene edit to be. But with the piggyBac system, and in fact it's been published, you can look for a TTAA site in the exon itself, because when it comes back out, it's going to be clean, and so it'll leave your exon intact, only with the gene edit remaining. So, so that's an important point. And so, even if there's not a TTAA site, usually you can find a silent one introduce a silent change in the exon itself. So it's very flexible. So again, we're going to put the selection cassette flanked by the piggyBac ITRs right at that close TTAA, preserving the genomic sequence. And in this case, since we're putting it upstream of the base edit, then the 3' homology arm is the one that will incorporate that base edit. So this is a picture of the kind of the finished product of and what the targeted locus will look like when you get correct targeting. It's a little busy, but hopefully it makes sense to you all. So again, you can see the endogenous TTAA that you saw in the upper pane right there. The red sequences are just restriction sites that are kind of conveniently located in the vector that I left in there. And then you can see the ITR. So the green is the one ITR, inverted terminal repeat of piggyBac. The yellow is the other. And you got the PGK Neo poly A in this one. In this case, I haven't shown the one with the TK gene, but obviously that is often used, and I'll talk about that in a minute. But so you can see that you've got a 5' homology arm, and that can be anywhere from 500 to 750 bases in general. You go right to the TTAA that's part of the genomic sequence, right into the piggyBac selection to the other piggyBac, and then you have to add another TTAA site. That purple TTAA is added in the vector because when piggyBac brings everything back out, it matches up those two TTAAs to leave just the one when it leaves. And then you can see the base edit. So in this case, we changed that G to a C, right where we indicated that we would. And you can see that it's close enough to the selection cassette where it should be incorporated into the homologous recombination event, and then your 3' homology arm. So I hope that's clear. You can chew on that a little while longer after the seminar if you need to look at it a little closer. But this is a typical gene edit type vector that we would make. So we're going to step now through. We're going to assume that we got correct targeting of our gene edit and our selection cassette, and now we want to get the selection cassette back out of there so that our gene edit remains in our gene. And the way we do this, as I mentioned earlier, is to use that thymidine kinase gene as a negative selection for any cells that still contain the cassette. And so what we do is we transfect the correctly targeted clones with piggyBac transposase. And we, again, like Jack mentioned, have a transposase that is excision only. And so we know that it won't reinsert, when it takes the selection cassette out, it won't reinsert it. In addition, you have the selection of ganciclovir or FIAU, which will kill the cells that have not lost the cassette. So they have to remove the cassette to live in the selection. So it's a good way to ensure that you're selecting clones that have lost the cassette, as well as selecting against any clones that may have gotten some random insertion of the TK gene, because they too will die. So if they haven't been removed, so this ensures that your final clones are cassette free. And now they'll be sensitive to the antibiotic now again, whereas before they were resistant upon targeting. Okay, so just going to go through a couple of examples that we've done in-house at Transposagen. This is very similar to what I just showed you, but kind of in cartoon form if it's a little easier to see. And in general, again, we've got the locus that we want to alter. The point mutation is indicated by the orange figure there in that exon. So we designed a TALEN to the very close to where we wanted the edit to occur, put in a PGK Puro TK cassette, and then targeted with that, and selected for those target clones with Puro at first, and then look for the ones that were correctly targeted with PCR products that one of the primers is outside of the arm of homology. And you do that on both sides. It's important to do the 5' and 3', screening in that manner, and then you can sequence those products and look to make sure that it's targeted like you want. Look for the gene edit was incorporated by sequencing those PCR products, and then again, using ganciclovir or FIAU, you can counter-select after you add the piggyBac transposase and get your final product, which is a footprint-free edit. One important point that is required here is to make sure that your TALEN or guide RNA binding and cutting site is at a spot where the insertion is taking place, because you don't want to be able to cut. You don't want to cut up your vector. So if you design a cut site that's fully in the vector, then the vector can get cut instead of just the genomic DNA. So that's one important consideration. And I've already mentioned the excision-only piggyBac. And this is, I want to show you just to illustrate other capabilities. Sometimes people want to do very precise small gene edits, and sometimes people want to do very big insertions, which is also possible. And this is one that we have done that involved knocking in a rather large piece of DNA, including an intronic sequence, into an existing gene, such that it would just be expressed in the same way as the gene is expressed, but in this case expressed this different sequence. And so in this case we put the selection cassette in the intron, flanked by the ITRs again, but right at an endogenous TTAA in that intron. And so we've designed two TALENs in this case, one to each end, to help ensure that the genome was cut at both spots, so that we could get this big piece of DNA in, and then selected for Neo. This is Neo TK. I just didn't draw the TK onto this particular figure, but and you can see the small little TTAA sites there, I hope, near the Neo. And then look for targeting. So we, again, like I mentioned, we use longer PCR products to look for correct targeting with primers that start in the insert and go past the arms of homology. And then you can sequence those products to see that, yes, in fact, it is correctly targeted. And then you take those clones and do the piggyBac excision, like I've mentioned. And this is just an example of, again, you can verify that the excision, not only that it occurred, but it occurred footprint-free as you expect, and that your allele is left in the exact manner in which you had planned it to be. And this is just a project that we've recently completed, and just to illustrate some of the capabilities that we have. And then I wanted to just take one slide to mention that obviously the alternative technology to put things in the genome and remove them and most popular, obviously, is the CRISPR system. There's also the flip thread system. And so, CRISPR obviously has been a great tool for research for a long time, but it does have its drawbacks, and a few of these are listed here. Obviously, cryptic LOX P sites are present in the genome and can produce off-target mutations. The figure to your right is just from a paper that shows, if expression of CRI in cells can lead to abnormal chromosomal issues. The column on the right shows that to be the case. So you don't want to, you can get unintended and unwanted mutations and unknown mutations potentially using CRI-LOX. In addition, in some cell types, you may get methylation of the LOX-P site, which can spread to surrounding sequence that might alter or silence the adjacent genes. And then, of course, the real, I guess, problem you might have with this system is the unknown effects due to leaving 34 base pairs behind when you're done. We can, despite your best bioinformatic efforts, we never for sure know if the place you're putting that LOX-P is an important spot, and you can't put it in an exon, which is something that you can do with piggyBac. And so piggyBac kind of gets around all these issues. It's known not to have these adverse effects on the genome, like cryptic LOXP sites do. There's no footprint, there's no scar left behind. So that's why we think that piggyBac is superior to the CRISPR system. And then again, just one slide to make the point that to do all this, you have to get the DNA in the cells, and we are perfectly capable of that. We've been doing it a long time, have the equipment to make that happen, and can transfect both obviously transformed cell lines as well as stem cells at whatever rate is needed to get the projects done. And then we're going to end this, move away a little bit from the gene editing part, and just to say that piggyBac is a really powerful and easy technology for just transgene insertion into cell lines, or make stable cell lines. It's non-viral, obviously. It's extremely efficient, and I'll show you a slide to illustrate that. But you can kind of titrate how many insertions you want to put in. A very large cargo capacity that's been published on, and so it doesn't have that limitation that a lot of technologies do. We have the ability to use a Splinker-Ret PCR to identify the integration site where the transposon inserts your transgene, so that you can know for sure where it is. And again, you can get it back out if you need to, so it's different than a standard stable cell line. You can put it in, if you need to take it out to then reverse your phenotype or whatever you're looking to do, you can get it back out and do the comparisons there. So I'll show you just one or two slides to illustrate that. This is a simple slide that shows if you put a transgene in with antibiotics resistance in with a piggyBac transposase, you get a lot of cells out the other side that have the transgene integrated, and it's stable. Lasts for many passages. You can, like we said, change the copy number as needed. And this is a final slide here just to show that from the literature showing that someone who needed to do kind of a more complex stable cell line where they needed it was a protein that had multiple components. They put all of those components in at the same time in the same cell line using piggyBac, and were able to show expression of this protein, activity of this protein, and that it was stable over many passages of the cells. So this is just a kind of a final capability that piggyBac has that we want to make you aware of, that you can either do in your own lab or we could do for you as well. So that's it for my part of it. I'll turn it back over to Jack and let him finish up and hopefully answer any questions I might have raised at the end. So thank you.
Thank you, Ken. I really appreciate that. The technical portion of our webinar. To conclude, I just wanted to talk about a couple advantages of partnering with Transposagen for these genome engineering, these cell line engineering projects. Transposagen is a high-quality, full-service genome engineering company from design to creation of any project, and we can tailor each project to fit the needs and the budget of the client or the project itself. We also offer free feasibility studies. So if you can provide us with some details of your project, we will design the tools and suggest the technologies that are required for creating that gene edit in the cells. And then we will also provide you with the information on the feasibility study as well as custom quotes and timelines, and that's completely complimentary with no obligation to move forward with the project. As we mentioned earlier, we have a very experienced genome engineering scientific team. We provide unlimited technical support, flexible milestones, which I'll talk a little bit more about, and importantly, we provide the customer with full ownership, and they maintain the intellectual property for the cell lines that we create and any discoveries that are made using these cell lines. Ken Miller is our project manager and senior scientist for cell line engineering projects, and Carlisle Endell is our director of custom animal models on the animal model side of things, both with plenty of experience in design and construction of gene engineering projects. So just a little bit of an overview of our custom cell line engineering services. We offer the transgenic or stable random integration cell line creation in transformed cell lines or stem cells using the piggyBac transposon system. We offer the knockout cell line using any number of different targeted nucleases, whether it's a TALEN, CRISPR, or any type of technology, and also the targeted knock-in cell lines where we will either create a donor vector or a footprint-free gene editing vector for a gene editing project. And so we also provide vector creation services. So if you have a project that you want to create a cell line, but you want to attempt the project in-house, we will create those vectors for you and deliver those vectors directly to your lab so you can have your specific application and cell line generation. So we have the TALENs, the CRISPR, both nuclease and nickase, will create donor vectors as well as custom footprint-free gene editing kits based on the multi-vector series that Ken talked about earlier. We'll also provide, design and create custom piggyBac vectors for stable transfection and cell line engineering, and we have kits for off-the-shelf piggyBac vectors where we will provide you with either the piggyBac hyperactive transposase, which will get your transgene into the cell at a very, very high efficiency, as well as a excision-only piggyBac transposase, and then the off-the-shelf transposons, and we offer the premium programs and activity testing. One other service that we offer is custom cell line activity testing. So if you have a cell line of interest that you have in-house and you'd like to either develop targeted nucleases that are going to be active in that cell line or have transposagen optimize that cell line for gene editing purposes, we can bring that cell line in-house and optimize the cell line for culture conditions, transfection, and gene editing for you, and then also do the gene editing projects as well. And when we send you the PDF of the slides, you'll have both the academic product list and pricing as well as the industry product and pricing. I mentioned that we break down all of our projects into milestones. This is just one example of how we break down cell line engineering projects into three milestones: vector design and creation, and we also provide the premium service, which means we do the activity testing of those nucleases. Milestone 2 would be cell transfection, typically cell transfection or nucleofection, and milestone 3, selection, screening, and delivery. We can change this to fit any project, so we've done 5 or 6 milestones as well, but this provides flexibility for the client. So at each milestone, we don't get paid until we meet that milestone, and at the end of each milestone, the client has the opportunity to either move forward with the project or terminate the project or bring in the vectors to create the project in-house, whatever method that they want to move forward with. And just as a summary, Transposagen has a diverse technology portfolio, as you can see from our summary up here, with these different projects that we offer, the different technologies that we have available, and the advantages of these technologies that Ken went over earlier. We have an experienced team for custom design and production, and we offer flexible milestones and pricing structure, the client maintains all the ownership and the intellectual property, and we believe that this all translates to successful partnerships. So with that, we will answer one question that we received. Ken, are you on the air here? We had a question. Ken, how many, when you're designing a donor vector for targeted knock-ins or footprint-free gene editing, how many base pair changes need to be made in order to ensure that the nuclease does not cut your donor vector, and only you ensure targeted integration for that project?
Yeah, so when I think I might have mentioned, if I failed to, one of the considerations you have to think about is obviously that if your XTN or CRISPR-Cas is going to cut the donor vector. And so what we like to do is design the TALEN to cut right very close to where that TTAA insertion point will be. And what that does is your vector has an insertion at that site. So, 2 kb insertion between where the forward or left TALEN will bind and where the right TALEN would bind. So in the vector sequence, even though the binding sequence might be present for each part of the TALEN, they can never be close enough to initiate any cutting. So your vector is fine in that sense, whereas the genome should be cut. So that's how we do it, and the TALENs are fairly, you can get pretty darn close usually to where you want to cut, and then with some kind of CRISPR-Cas system, you would just make the portion that binds the gRNA be also bisected as close as possible by that insertion point, so that the vector can't be cut.
Okay, I have one more question for you. When working with an essential gene and wanting to make a single mutation, the piggyBac cassette would need to be outside of the gene and keep the promoter sequences intact and functional during the intermediate step. Is that correct?
Yeah, so that would be a consideration, and yeah, so I mentioned that you could put the insert or the selection cassette in the exon if you needed to, but that's a good point. If you're working with an essential gene, and hopefully the biology of that gene is known going into the project, you would want to put it in the intron somewhere so that hopefully correct splicing could still occur while your selection cassette was in there. And then that's if you want to do biallelic editing, of course. I mean, if you just need one allele change, that wouldn't be as big a consideration. But yeah, so you would, in that case, and if you didn't know whether your gene was an essential one for whatever reason, you might want to just default to putting the cassette in the intron when possible. That way, you would avoid those kind of issues.
Okay, thank you. And on the other end, I just wanted to mention, I don't know if we went over this in detail, but the beauty of having piggyBac as footprint-free as opposed to the CRE-LOX system or other systems is that if you have a 1 exon gene or a huge exon that you want to change a single base pair, or you want to make some change in the middle of that exon, you can actually target within the exon with piggyBac and then remove it since it's footprint-free. And with other technologies, you would not be able to do that because, of course, you would leave the LOXP site within the middle of the exon. So you are basically forced to create a huge donor vector with large arms of homology where we can go right into the exon because of the footprint-free nature of the technology. So with that, I'd like to conclude the webinar. Thank you to all our attendees. And if you have any additional questions, please contact either me, Ken Miller, and if you have any projects that you're interested in and you want a feasibility or a custom quote, please let us know, and we'll get back to you as soon as possible. Really appreciate and thank you again for joining us today.